Hello guys, in this block of the subject we are going to talk about the structure and the function of the salivary glands. That means to talk about its anatomy, knowing what they are located and its relationships. We are also going to talk about the histology, so we know the inner structure of the salivary glands, and we're also going to talk about its physiology, regarding its innervation and its production. The salivary glands are exocrine glands located around and within the oral cavity. Its function is to produce the saliva. This saliva will be in charge of helping the deglutition process of the food, its disgregation, to dissolve some elements, and it will be the beginning of the chemical digestion. It's also going to act as a helper to the immune system and defend against microorganisms. It's also going to maintain the oral pH, the mineralization of the enamel, and generally speaking, the maintenance of the oral mucosa. We can classify the salivary glands accordingly to its functionality and its size. Thus, we can talk about the major salivary glands, also called the principal salivary glands, or we can talk about the minor salivary glands, also known as the secondary salivary glands. Examples of the major salivary glands would be the parotid gland, the submandibular, and the sublingual glands. As the minor glands, they are scattered all over the oral cavity, and we will find it within the tongue, the palate, the lips, and the cheeks. Another way to classify the salivary glands is accordingly to its secretion. Then, we can define them as the serous type. The serous type of salivary glands produce a very liquid secretion. In this group, we are going to find the von Eppner glands surrounding the circumvallate and the foliate papillae in the tongue, and the parotid gland. Then we have the mucus gland, which produce glycoproteins and mucins. Actually, we will not find pure mucus salivary glands because they always have a certain number of acini, which are both mucus and serous. Example of mucus type will be the sublingual and some of the minor salivary glands. Finally, we have the mixed salivary glands, where you can find acini, which contain both serous secreting cells and mucus secreting cells. A very good example of this type would be the submandibular gland. Now, let's talk about the major salivary glands. This type of salivary gland, they have a very obvious volume. Two of them will be located outside of the oral cavity, which are the parotid and the submandibular glands, and one of them is within the oral cavity, which is the sublingual. In any case, the three of them uses several major ducts to get into the main oral cavity. Now, regarding of the parotid gland, it is the major of all of the salivary glands. It has a large volume and it weighs about 25 grams in average. It looks like lobulated with a yellowish gray color, and it contains a large amount of adipose tissue. This adipose tissue is going to make it flexible and able to adapt to the movements of the mandible. The parotid gland, it is covered by the parotid shed, which derivates from the deep cervical fascia. It is located by the sides of the face in a region called the parotid zone, which presents an irregular shape, which depends on the mandibular movements. The parotid gland, it is divided in two large portions by a fibrous tissue in where we can find the infraparotid plexus of the facial nerve, also called the facial plexus, which originates the superficial and the deep portion. Now, let's have a look to the parotid gland relationships. In the anatomical point of view, the parotid cell has an irregular shape of a prism with three sides, two extreme points, and three borders. Now let's define the relationship and the surroundings of the parotid gland, and also we are going to comment on the structure which are going to cross this major salivary gland. Thus, we can say that there is a lateral or external side which is related to the parotid fascia, which derives from the deep cervical fascia and is closer to the skin. There is also an anterior, also known as the anterior internal side, which is related to the posterior border of the masseter muscle, the mandibular ramus, and also with the medial pterygoid muscle. Finally, there is a posterior side, which is also known as the posterior internal side. At this point, the parotid gland from the lateral to the medial direction, it is related to the following structures. First, we can observe the sternocleidomastoid muscle. Then we have the gastric muscle posterior valley. Close to this valley, we can find the internal jugular vein and the styloid process. And of course, those muscles which use this process as the origin for its insertion, such as the stylopharyngeus, the stylohyoid, and the styloglossus muscles. The parotid gland, as it contains three sides, it also means that it contains three vertices. The anterior vertex is related to the masseter muscle, and it is the place where the parotid duct arises. Then we have the medial or internal vertex, which is related to the oropharyngeal muscles. Finally, there is a posterior vertex, which is closely related to the sternocleidomastoid muscle. Just to finish the relationships of the parotid gland, we can say that superiorly contains an extreme point which is related to the auditory canal at the posterior part and the temporomandibular junction at the anterior. Regarding the inferior extreme, it is related to the mandibular angle, the submandibular gland, from which it would be separated by a fibrous septum called the submaxillary or submandibular band. Now, let's talk about of those structures which crosses the parotid gland. They are most important as they give the blood supplies to the face as well as the innervation. One of the most important structures that we will find will be those branches of the seventh cranial nerve, that is the facial nerve, which are going to be those terminal branches with motor functionality to give the innervation and the action for the facial muscles so we can make facial expressions. We will not further describe the many branches of the facial nerve here as we have a dedicated chapter for this purpose. Another important structure that we have here, it is a branch of the V3 mandibular nerve, which is called the auriculotemporal nerve, and which is going to leave branches with sensory and motor information to this region. Now, regarding of the blood vessels, possibly one of the most important, it is the external carotid artery, which penetrates into the parotid cell, and from there arises its large collateral branch, the posterior auricular artery, and a little bit more superior, it is divided into its terminal branches, the superficial temporal artery and the maxillary artery. As for the venous drainage, within the parotid cell, we have the confluence of both the maxillary and the superficial temporal veins, which originates the retromandibular vein. Then, part of the retromandibular vein is going to drain into the external jugular vein. This external jugular vein is going to exit the parotid cell through the inferior extreme point. Finally, we have the lymphatic drainage. Associated to these lymphatic vessels, we have the corresponding lymphatic nodes, which are also known as the parotid nodules. Then we can find the suprafascial ones, which are superficial to the parotid fascia. Then we have the subfascial ones, which are lying between the fascia and the lateral axis of the gland. Then we have the deep interglandular parotid nodules in the thickness of the gland, located around of the external jugular and of the retromandibular veins. The parotid gland is fairly a part of the oral cavity proper. Therefore, it needs assistance to get the saliva into the oral cavity. This assistance is being provided by a major duct called the parotid duct, also known as the Stensen duct. The parotid duct it is very wide and it has thick walls. It arises from the anterior border of the gland, and from there it is going in an anterior direction through the lateral side of the masseter muscle. Once it gets to the anterior border of the masseter, makes a turn of almost 90 degrees, is going to surround anteriorly the ball of his shaft, and then it's going to pierce the vaccinator muscle at the level of the second molar of the maxilla. In this very point, it's going to open through the oral mucosa into the oral cavity to deliver the saliva. The parotid duct it is closely related to the transverse artery, as both of them follow the same trajectory. Surrounding the parotid duct we may find another structure which is sometimes called as the accessory parotid gland. In some specimens, it is fused to the main parotid gland as a process, and it has the same functionality. Now, let's talk about of the submandibular gland. It is located in the suprahyoid region on the lateral part. It is located between the anterior border of the sternocleidomastoid muscle and the anterior belly of the digastric muscle. It is also anterior to the mandible's angle. It will be lying in a depression of the internal part of the mandible and lateral to the suprahyoid muscles, and also of the lateral root of the tongue and the pharynx. To this region it is known as the submandibular zell. The submandibular gland is smaller than that of the parotid, and its weight is about of 7 grams. The submandibular gland can be divided into two portions. Thus, we have the superficial portion, which is the largest. It goes in an anterior direction, following a plane underneath of the mylohyoid muscle. The lateral part of the superficial portion, it is accommodated into the internal part of the mandible in the region called the submandibular fossa. Then we have a smaller portion, which is called the deep portion. This deep portion goes around of the posterior border of the mylohyoid muscle, and it penetrates into a little pocket which is made of the mandible, the mylohyoid muscle, and the hyoglossus muscle, which is called the submaxillary hiatus. Once in the hiatus, it goes in an anterior direction, above of the mylohyoid muscles, and lateral to the hyoglossus muscle. Its most extreme anterior part can contact with the posterior border of the sublingual gland. You must remember that the portion which is located closest to the skin, it is always the superficial one. When you look to the picture, the inferior part of the gland, it is the closest to the skin, therefore that is the superficial, the largest lobe of the gland. The one which is located closest to the tongue, which is more superior, it is closer to the oral mucosa and of the tongue. This is sometimes wrongly confused because you are used to work within the oral cavity, but that is meant to be the deepest part of the oral cavity. All in all, what we have is the following thing. The submandibular gland is divided in two portions. The one which is inferior, it is the superficial portion, also known as the superficial lobe. The one which is closest to the oral cavity, oral mucosa, it is the superior one, but it's also called the deep portion or deep lobe of the submandibular gland. Now, let's have a look to the submandibular cell. We can consider that it has the shape of a pyramid with a triangular shape with three sides, three borders, and three extreme points. There is a superolateral or superexternal side which is related to the submandibular fossa of the mandible. In this side, the submandibular gland, it is related also to the medial pterygoid muscle at the posterior part and also to the facial artery. Then we have an inferolateral or inferosternal side, which is covered by the superficial lamina of the cervical fascia, the platysma, and the facial skin and vein. We cannot appreciate this side in the pictures because it has been removed, but it is where the arrow it is pointing at. Finally, we have the medial or internal side. In its most anterior part, it is related with the mylohyoid and the digastric anterior valley. It has also relation with the hyoglossus and the stylohyoid muscles by the posterior part. At this point, the facial artery surrounds the gland to end by the superolateral side. The submandibular canal appears in this side. The submandibular cell, which is triangular in shape, has three borders. The lateral border will be the lower edge of the jaw. The inferior border will be the superior border of the hyoid bone, actually the greater horn, and the thyrohyoid membrane. Finally, the top border will be the mylohyoid muscle and line, with its insertion in the jaw. There is an anterior end by the anterior belly of the digastric muscle, and there is also a posterior end, which is the submaxillary septum that separates this gland from the parotid and the superior constrictor muscle of the pharynx. The submandibular gland, it is apart from the oral cavity proper. For this reason, it needs a duct to get the saliva into the oral cavity. Thus, we can talk about the submandibular duct, also known as the Wharton's duct or submaxillary duct. It is about 5 cm long and fairly thin compared to that of the parotid gland. It is going to originate from two main collectors from the medial or deep side of the gland. From its origin, it is going to orientate in a trajectory oblique in an anterior and medial direction. It is going to get together with the lingual vein, the lingual nerve, and the hypoglossal nerve too. At the beginning, it's going to be medial to the lingual nerve, but a little bit after, it's going to cross it, so it's going to end up a little bit more lateral. The Wharton's duct is going to end up by the oral mucosa by the side of the frenulum in a structure called the papillae. The last of the major salivary glands, it is the sublingual gland. It is the smallest one, weighing no more than 3 grams. The sublingual gland, it is the most anterior of the major salivary glands. Roughly, it is located anterior to the submandibular gland, underneath the tongue and under a mucous membrane of the floor of the mouth that we call the sublingual fold. There are several features of the sublingual gland which makes it different from the other major salivary glands. One of these features it is that it's not an extra oral gland because it's right underneath of the sublingual fold, therefore it's properly located by the oral cavity proper. Another feature is that it has an underdeveloped capsule. In addition, it doesn't have complete septa. Thus, it's more like a set of glands that get together, but they contain independent conducts to get into the oral cavity, which is something that does not happen in the parotid gland or the submandibular gland, where we only have one major duct to get into the oral cavity. Now, let's talk about of the location and the relationships of the sublingual gland. Regarding its location, it is lateral to the Wharton's duct, the lingual artery and nerve, the genioglossus and the geniohyoid muscles. It is superior to the mylohyoid line and muscle, and is medial to the sublingual fossa of the mandible. We could also say that the sublingual gland contains two sides, the lateral by the sublingual fossa and insertion of the mylohyoid muscle, and the medial, which is the one related to the genioglossus muscle and the longitudinal muscles of the tongue, the submandibular canal, and the lingual nerve. We can also consider that the sublingual gland has a superior border related to the alveololingual group, which at this point forms the sublingual fold. The inferior border it is associated with the geniohyoid, and there will be an anterior border towards the mental symphysis and the contralateral sublingual gland. The sublingual gland, to deliver the saliva into the oral cavity, it does it through several excretory ducts, in numbers from 8 to 20, which are also known as the revinous ducts. At the anterior part, we have a larger sublingual duct, also known as the duct of Bartholin, which joins the submandibular duct to drain through the sublingual carbuncle. Now that we know the anatomy of the major salivary glands, let's have a look to the inner structure of those glands. Generally speaking, the saliva is being produced by a structure which is called the acynus. Several acynus together make either the minor or the major salivary glands. We can differentiate several types of acynus depending on the type of saliva which is making. Thus, if the saliva which is making is aqueous, we say that it is a serous acynus. Regarding the major salivary glands, the only pure serous secreting gland it is the parotid gland. Then we have a type of acynus which secretes a saliva which has a large number of mucus. Therefore, we call them mucus acynus. Finally, there is a type of acynus which has cells which either secrete mucus containing or serous containing saliva. This will be the mixed acynus. This type of acynus, the mucus and the mixed one, we will find them in different proportions in the sublingual and of the submandibular gland. Thus, the submandibular gland is rich in serous secreting acini, compared to that of the sublingual, which in the contrary it is predominant in the mucus secreting acini. In the major salivary gland, there are groups of acini which get together and separated, forming something that we call the lobule. All the acini within a lobule are connected one to another with something that we call the intralobular ducts. There are several types of them depending on the caliber. The smaller ones are called the intercalated ducts, which are directly connected to the acynus. Then we have the striatic ducts. Once we get outside of the lobule, we have the interlobular ducts. This one, they have a very large caliber, and in some occasions the walls get very thick with several layers of cells compared to those intralobulars which are normally a single layer of cells making the duct. Now let's have a look to the histology of each one of the major salivary glands and a few details of those of the minor salivary glands. The parotid gland has the following histology characteristics. One of them is that it's being contained with a thick capsule and it contains well-defined septa which divides the gland into lobes and lobules. Another feature is the large amount of adipocytes. Much of the parenchyma can be replaced by stroma, reaching this adipocyte by aging. The parotid gland, it only contains serous-secreting acini. This saliva is very rich in amylases, but it also contains certain amount of sialomucins and sulfomucins. In this lobe magnification picture, we can observe the following features. The pale pink staining represents the septa. Then we can differentiate the very thick and large septa which divides the gland into lobes. Within the lobe, we see very thin septa which create the lobules, and each lobule is going to contain the acini. As for those regions without the staining, are representing the adipocytes. In these two pictures, we can observe the parotid gland with higher magnification. In the left-hand side picture, we can clearly see those cells without staining and white color which represent the adipocytes. Then there is little round structure with a very dark pink color which represent the acinos which are scattered all around. In between them, we can see the intralobular ducts. Some of them are larger than others. Let's get into the right-hand side picture to see everything in even higher magnification. Thus, with the yellow and the red colors, we can observe several acini together. It is not unless we get into even higher magnification when we can observe independent acinos. Something that we can observe below of the yellow circle is a large intralobular duct. In this case, we can see that there are several layers of cells making this duct. Now, let's get into even higher magnification to see a single and independent acinus. In this picture, we can observe in very high magnification individual acinus, which are highlighted here within the yellow circles. When we have a close look to the acinus, we can observe several types of stainings. The first thing is that we can see areas with very pink light color. These are the divisions in between several cells. Then, all around we can see very dark dots scattered all over the place. These are the vesicles with the serous secretion. Another feature of the serous acinus is that the apical region doesn't show the lumen. It is not that it's not there, it's because the apical region of these cells intermingle one to another, blurring the duct within the acinus. We can also observe the intralobular ducts. We can distinguish them because in this case the cytoplasm of these cells have a pale pink staining and the nuclei, rather than being in the periphery, they are scattered through the middle of the cells. In this picture, we can observe with number 2 the smallest caliber of the ducts which is directly connected to the acinus. This is the intercalated duct. With number 3, we can see a very well-defined lumen surrounded by cells which are proper of a duct. This is the striated duct which has larger caliber. Finally, with the number 4 and no staining at all, we have the adipocytes. Now, let's talk about the histology of the submandibular gland. In the same way as the parotid gland, the submandibular gland contains a well-developed capsule. It has a structure well organized of parenchyma and stroma, with interlobular and intralobular septa. The submandibular gland is a serous mucous tubular gland. This means that the shape of the acynus is tubular. In addition, the tubule contains several cells capable of making serous secretion and another type of cell which are mucus secreting cells. For this reason, the submandibular gland it has a mixed component of salivas. However, we have to say that the number of acini is 10 to 1 serous to mucus, and therefore it is predominantly a serous secreting gland even if there is certain amount of mucus saliva. The acynus have a peculiar structure that we shall see, and it's called the Giannucci demi-moon. Thus, we shall see that the tubule is mainly made of mucus-secreting cells. However, it's going to have a cap of cells which are serous-secreting cells. That's what it makes the demi-moon. Regarding the content of saliva, not surprisingly, is very rich in mucin such as glycoproteins. Now, let's have a look to a micrograph of low magnification of a section of a submandibular gland. As you can observe, it's very similar to the one of the parotid gland, with one exception, which is the absence of adipose tissue. We may need to go into higher magnification to detect the adipocytes. As for the rest, you can see that it's very similar to the one of the parotid gland. Thus, we can see the connective tissue with the pale pink staining, making the septa and dividing the gland into lobes and lobules. In addition, in this picture, we can see a very large caliber interlobular duct. Then, within the lobule, we can see several intralobular ducts. If we have a look to the submandibular gland with higher magnification, as we have in this picture, the first thing that we can observe is the following thing. In the left-hand side of the picture, there is a number of well-stained acini, which have diffuse serous secreting acini. In the right-hand side of the picture, on the contrary, we can see there are several acini with poor staining. That is because the staining doesn't go into the mucus-producing cells. Therefore, what we have in there is a mainly mucus-secreting acini. Getting into the details of the acini, we can observe with the yellow dot circle a single serous secreting acinus, which contains the dotted vesicles and the basal nuclei. The acinus with the red dot circle, it is represent a mixed acinus. You can observe that in the top of the tubular structure we can see a well-stained set of cells. That is the demi-moon with the serous secretion. Down to the tube we can see that the majority of the cells have very poor staining. These are the mucus-secreting cells. We can also observe several striatic ducts that contain a very even staining in the cytoplasm's center nuclei, and it is the one structure highlighted with the blue dot circle. In this case, we are seeing several striatic ducts together. Finally, in the top left-hand side of the picture, there is an empty white space which is possibly representing an adipocyte. Finally, let's have a look to the histology of the sublingual gland. The sublingual gland is not a simple pair of glands in each side of the oral cavity. However, there is a major gland with several minor units with independent ductal systems. As we have said, the septa it is not clear. and the type of acini will be both mucus and serous with predominance of the mucus one. We are also going to observe the serous cap, which is going to make the mixed acinus. Now let's have a look to a micrograph at low magnification. In this picture we can observe that the amount of staining is being reduced, and therefore we can say that there is a predominance of the mucus secreting acinus, which, if you remember, the mucus cells do not get very well the staining, and that's the reason why the picture is more clear. With the yellow dot circle, we have an independent acinus. You can observe that the center of the acinus has no staining. This is because we are observing the mucus secreting cells. In the periphery, we can see a better staining, especially in the lower part. This is because we are observing the serous secreting cells, which makes the cap that we call the demi-moon. The two other features that we can observe is the connective tissue making the septa. In this case, it's very difficult to recognize whether we have lobes or lobules because the septa it is not very well defined. The other feature that is easy to observe it is the interlobular ducts. We can observe two of them by the right hand side of the picture with a very large caliber and surrounded by connective tissue. Finally, let's say a few words regarding the minor salivary glands. The minor salivary glands are distributed within the oral mucosa and submucosa. They are very small and very numerous. They are located very close to the inner surface of the mouth and connected by very short ducts. We may find them all around except of the masticatory mucosa in places such as the gums or medial and anterior parts of the hard palate. On the contrary to the major salivary glands, the lobule subdivision is not always complete. Regarding the type of secretion, all of the salivary glands have a predominance of mucus secretion, except for one minor salivary gland, which is called the von Effner gland. This von Effner gland is located in the dorsum of the tongue, and together with the parotid gland, it is the only one which contains 100% serous secreting cells. Thus, most of the minor salivary glands resemble in general to the sublingual gland. These minor salivary glands contain mostly parasympathetic innervation. The function of the minor salivary glands it is a protective function of the teeth and mucosa. We have several types according to its location. Thus, we may have the labial, the genian, the palatine, and the lingual. Within the lingual, we are going to have the Blandings and the Nun's glands, the dorsal posterior Weber's glands, and the von Effner glands. In the next slides, you have the principal characteristics of each one. Once again, thank you for your attention